Anyway, our next session is PFAS trophic transfer in freshwater aquatic systems, injury determination methods for natural resources with Dr. Jeff Morris. Jeff is the senior ecotoxicologist and co-director of NRDAR Consulting, Biodiversity Research Institute. And Jeff has been supporting trustees with natural resource damage assessments for over 15 years and conducting ecotoxicology and bioremediation research for over 25 years. He's an ecotoxicologist and aquatic biologist at Biodiversity Research Institute with expertise in field and lab monitoring and bioassay, uptake and toxicological modeling, and injury determination and quantification. Jeff has a PhD in zoology and physiology. Welcome, Jeff.
Thanks, Christian. Can you see my slide okay?
I see it. Great. It's very clear. Thanks.
Good to go. All right. Alrighty, let me get going here. So first want to acknowledge co-authors on this project. There's a bunch of them, lots of colleagues here at BRI and some former colleagues who have been involved in different aspects of what we're going to discuss today. Probably not a shock to a lot of people, but just also kind of want to just mention confidentiality. I'm not going to divulge anything that's confidential, but because I can't do that, there will be some details in some of what I'm presenting, including locations and some other stuff that it just can't be shared at this time. So kind of an overview on what we're talking about. So we've been, for different NRDA projects that we've been working on, we've been developing a modeling method to allow us to predict tissue concentrations in higher trophic level organisms based on concentrations of contaminants in sediment, water, or prey species. So, this is a model that could be applicable for looking at uptake and exposure modeling, but then also to potentially determine tissue concentrations in organisms and then apply different types of tissue concentration thresholds or even dietary exposure thresholds to those prey species. Just 1 second here. Okay, so trophic magnification, why is it important for assessing injury to natural resources? Because big things eat little things, and then those contaminants increase in concentration as they move up the food chain. I'm sure most people on this call understand the concept of biomagnification. So when we're thinking about trophic magnification, it's just trying to quantify that through different trophic levels. So in terms of trophic levels, we got trophic level number 2 or trophic level 2, which would be a primary consumer. That's something that eats plants, all the way up to trophic level 4 or 5 or higher, depending on where things fall. So you can kind of use this concept along with tissue chemical concentrations and do a regression. And what you typically see with a lot of these contaminants is that as you move up different trophic levels, those concentrations get higher and higher. That's biomagnification. And if you fit a regression to that, the slope of that line is the trophic magnification slope. So that tends to be steeper in contaminated habitats. So that's a key component to some of the modeling work that we've looked at. And the concept isn't new. So this has been around for a while, especially with folks that have been studying mercury and methyl mercury. This is a figure, sorry, this is a fuzzy figure, but it's actually fuzzy in the publication also. But this is some of Matt Chumtal's work looking at methyl mercury accumulation, different organisms at Caddo Lake in Texas. So the same idea, you can plot tissue concentrations for different critters on the y-axis, and when you do this, it's always log-based concentration. So this is for methyl mercury, the log of methyl mercury versus the trophic position or trophic level for these different organisms. Again, you can fit a line to that. The slope of that line is your trophic magnification slope. So like I said, a lot of work's been done with this and mercury, also PCBs and PFOS, which I'll be talking about here in a few slides. So in terms of trophic magnification slope, as I mentioned before, the steepness of that slope is important, indicates uptake rate into the food web. But this is just for tissue concentrations. There's no tie back to any kind of sediment or water concentration with this particular method. You may be familiar with something called a bioaccumulation factor, which is essentially just looking at the concentration of a contaminant in a tissue versus the concentration in the medium that they're exposed to. So it could be tissue concentrations in sediment, tissue concentrations in water. There's a variant of that that uses lipid normalized tissue concentrations and organic carbon normalized sediment concentrations. But what these are all essentially trying to do, whether it's just a ratio or trying to do some kind of regression, is to look at how what that relationship is between the exposure media, which might be sediment, and the concentration in a given species. And that's useful. But if we want to think about potential exposure to higher trophic level organisms, this isn't always the best way to try to estimate that. Also, because those organisms may not be in close contact with the sediment, even though there's a direct link through the food web. So what our model is built to do is to kind of take both of these concepts, trophic magnification slope, bioaccumulation factor from a given media, combine those into a model that can take media concentrations, so sediment or water concentrations, and predict tissue concentrations at higher trophic levels, given the concept of trophic magnification and bioaccumulation from the media. So just kind of go through a few aspects of the inputs for the model. There are trophic levels that need to be assigned. If you're using, in some of the examples I'll show, using sediment data, you have to have a sense of what that spatial sediment data looks like so that you can confine that a little bit into specific areas that you use to calculate the trophic magnification slope. And then I'll show kind of some slides on how you generate that model and what the output looks like. So the first thing are trophic level assignments. So this is an important piece of this. Obviously, there's different ways to get to trophic levels. Kind of the gold standard would be using stable nitrogen isotopes. And there's a couple of things you can do with that. If you have that type of data for all of the tissue samples, you can just use that directly as your x-axis rather than a trophic level. But if you don't have it for everything and you want to calculate trophic levels, there are some different publications out there with different approaches to doing that. But essentially what you're doing is you're taking the stable nitrogen isotope signature for some kind of a baseline organism that's usually, say, a snail or a bivalve, and then assigning that with a trophic level of 2, and then looking at stable nitrogen isotope data for other organisms, using that to scale and come up with calculated trophic levels. For a lot of NRDA work and other work, there just isn't a lot of good stable nitrogen or stable isotope data for nitrogen, and therefore you have to do some other things. So if you're working with fish, there's a website called fishbase.org that has a whole bunch of information on different types of fish species. For most of those, they do report a trophic level that's based on dietary studies. And so it's kind of a good starting point. It's never going to be exactly the same as your site. We know that from comparing, actual data that we've collected with what's on FishBase. But I think FishBase does a good job of showing the relative difference in trophic levels between different species. And then sometimes you're not going to have any information like that that you can really quantify. So you're going to have to base their trophic position for, say, invertebrates or other species just on what you know about where they are in the food web and what they feed on. And honestly, for a lot of our work, we end up using kind of a hybrid approach with all three of these, depending on what kind of data we have and what kind of data we don't have. But one way or another, you need to get to trophic level assignments for all the tissue concentrations. Then if you're using sediment, you have to know something about that spatial coverage of sediment concentration. And, we can do something where we interpolate sediment concentrations and then assign kind of a grid cell pattern Or in an example that I'm going to show in a little bit, you can also just use a distinct lake as a study site, but you need to constrain that because you want to have sediment concentration from a given geographical area along with tissue concentrations from that area, and try to get that as as small as you can, so that that what you're looking at is tissues that have been exposed to those sediments and higher level organisms that have been exposed to prey species that have been exposed to the sediments. So once you have that, you can pull data from a given area. So in this example, from a particular grid cell, we get a sediment concentration that we'll use a little bit later in a later step in the modeling process. But we'll pull all the tissue concentration data from that site and put together a trophic magnification slope. This is trophic level for different tissue types and the tissue concentration on a log base 10+1 scale. So the slope of this line is our trophic magnification slope, and we also use the intercept from this as two key components to working on with our model. So what we do with that for a particular site is plug that into a regression for trophic magnification slope and for intercept based on sediment concentrations. So in this case, this graph represents one distinct geographical area or study site, but you have to have multiple sites in order to be able to fit a regression in this next step. So in these graphs, all of these points represent actual geographic locations or study sites for which you can develop a slope, and you have a sediment concentration, which is on the x-axis, or the intercept for that curve, again, versus the sediment concentration for that site. Once you have that for enough sites, you can fit another regression line and use the equation from that line to determine what your intercept should be or what your slope should be at a given sediment concentration. So we'll walk through the steps a little bit more, but ultimately what you get out of this once you're able to get to this point is you can derive a model that will predict tissue concentrations for different trophic levels. So each of these black lines are different trophic levels, and look at tissue concentrations at different sediment concentrations using that modeling approach. So you can see for some of the higher trophic level organisms, like a trophic level 3 or 3.5, you have a steeper slope than you do for some of the lower trophic level organisms. So that biomagnification is showing up there. You've got increased concentrations as you have higher sediment concentrations. And then you can, these dashed lines are meant to be, examples of thresholds or adverse effects concentrations you might have. So you can start to ask questions about, at a given sediment concentration, am I exceeding a particular threshold for organisms at these different trophic levels? And begin starting to think about injury determination and quantification using that type of an approach. So I do want to talk a little bit about some specific example with PFOS. And this is for a large area study that we did over the course of a couple of years in northeastern US. For this, the sources of PFAS, probably potentially atmospheric for the most part, but there might have been some also some local inputs. And for our study areas here that we use for the modeling, we actually had collected samples from 6 distinct lakes. So we have 6 lakes. We collected samples, analyzed all those samples for a suite of different PFAS. What I'm talking about here, what I'm focusing on, are the PFOS concentrations, and PFOS is definitely a PFAS that is known to biomagnify. So from each of those lakes, we have water, surface water, sediment, and biota samples. For the biota that we sampled, we have stable nitrogen isotope data for most of the samples, and we tried to collect samples that represent a range of trophic levels across that food web. So we have aquatic invertebrates, including bentho-macroinvertebrates, snails, and crayfish. For some of the lakes, we do have frogs, a lot of different fish species, so bass, various sunfish, suckers, and catfish, and then some loon tissue. So we do have loon blood samples from adults and chicks. We have some tissue samples from loons that were deceased or euthanized because they'd been attacked by an eagle or something else. We didn't sample that for this project, but it was just kind of opportunistic. And then there's also a lot of loon egg data because loon nests frequently get abandoned for different reasons. And so some of those eggs don't hatch, and you can collect those and look at the concentrations in the eggs. Because we were focusing on loons as a top predator, the fish samples that we collected, we kind of limited the size range there to not be larger than the size that you would have an adult loon typically feeding on. A little bit of a shout out to BRI. BRI was actually started a little over 25 years ago, and some of the first work that that was done at BRI and continues to be done now was actually capturing loons and collecting samples, primarily looking at concentrations of mercury in loon blood. But the method for collecting them is pretty cool. The loons are territorial when they're nesting. They have distinct locations on lakes that they protect and hang around in. And so you can go out at night and spotlight the loons, collect them, capture them, and get blood samples from adults. And then, like I mentioned, also from chicks, if the chicks are large enough. Okay, so kind of this trophic magnification model process. We have our 6 lakes. So this would be lake 1. For that particular lake, we look at tissue concentrations and trophic levels. We get a slope and an intercept, kind of like we talked about before. So for the actual lakes that we looked at, I have a couple examples of that. So this first one is our reference lake. We have invertebrate samples, which are these gray symbols, some frogs, which are blue, fish in green, and then over here we have loon data. So the red diamond is adult loon blood, and there's another one right behind this white symbol, which is an abandoned egg. And then we also have chick blood. I put that on here just to kind of see the difference, even though this isn't a log scale on the y-axis. But I'm not including the chick blood in this regression because they're not really sure without stable nitrogen isotope data, which we don't have for these samples for the loons, where they fall in terms of trophic level. They're definitely going to be less than the adults. The eggs would be the same because the eggs are a reflection of the tissues in the adults, and the eggs haven't started feeding yet. These chicks, once they're captured, and they might be 6 to 8 weeks old, and they will have been feeding on smaller, lower trophic level organisms. But anyways, for this particular example, this is our reference. Surface water concentration on average was 0.28 nanograms per liter. Sediment was 0.09 nanograms per gram. And then when we fit a regression here, we get our equation, and the trophic magnification slope rounded off is this right here. So it's 0.58. Compare that to one of our contaminated lakes, which I've got another example of here. Instead of a trophic level of 0.58 or a trophic magnification slope of 0.58, we have a slope of 0.77. So that's a steeper slope. The surface water concentration was 1.47 and the sediment was 0.25. So much higher than our reference lake. And again, kind of the same idea. We have loon blood, loon eggs, fish, and invertebrate tissue from this particular lake. And we did this for 6 lakes in total. So once we have that, we can kind of take the next step in this process, which is to plot up the trophic magnification for all 6 lakes versus the sediment concentration for each one of those, and do the same thing for the intercept here. So if we look at what that looks like for our 6 lakes, you get these kinds of relationships. So this one up here is sediment. This is trophic magnification slope versus the sediment concentration for that lake. And you see you've got a relationship here. You can fit a curve to that, use that equation to get to your trophic magnification slope and intercepts for given sediment concentrations. You can do that in this case. It doesn't always work out this way, but in this case, you can do the same thing with surface water concentrations. And then also, just to kind of show folks how it looks, I also plotted a trophic magnification slope versus the concentrations in the fish species. This would be average for the fish species that was collected. So in this case, we've got some options. We typically use sediment and or surface water to build a model, but you can kind of see that you've got some relationships here that allow you to predict trophic magnification slope based on concentrations in sediment water or prey species. So in the next step in this process, then you can choose a target sediment concentration that you want to model, use the equations for the trophic magnification slope here, and the intercept here, plug in your sediment concentration, and that allows you to predict what that trophic magnification slope is going to look like at a given sediment concentration. So, and I know there's a lot of subscripts and math here, but the end goal here with the modeling is to get a tissue concentration for a given trophic level, and this graph here would represent what that relationship looks like at a given sediment concentration. So you can then take that and look at different sediment concentrations and start to build something that looks like this, which we looked at a previous slide, that gives you a sense of what the concentrations at different trophic levels would be for that given contaminant. And then again, start to overlay different thresholds, whether those are tissue thresholds or maybe dietary thresholds for lower level organisms that might be prey species. But look at sediment concentrations. You could do the same thing in this case from what we've just been discussing for surface water concentrations, and I'll use those to predict what those concentrations look like in the higher trophic level organisms. So last slide here, just to kind of wrap up. We've used this approach, and it does seem to work well for trying to predict tissue concentrations across a continuum of trophic levels at given sediment concentrations. If you have good relationships like we have with this PFOS data, you can also look at those concentrations compared to predict tissue concentrations given certain surface water concentrations, and then compare those to thresholds and different adverse effects levels. And in terms of using this for assessment, it's a good tool when you have incomplete data, because you may know that there are certain organisms that are in a particular habitat that you don't have data for, but if you know what their trophic level is or can predict their trophic level, then you can use a model like this to predict what those concentrations would be in those organisms. And that's very important when you're thinking about higher trophic level organisms that may be difficult to collect. It's usually easier to get a water or sediment sample, or maybe collect some small fish rather than trying to go out and do collections of top level predators. And then the last thing is you can use this model to predict what the tissue concentrations will look like in the prey species for those predators, and start to think about potential effects from that aspect as well, which for any kind of assessment ends up potentially being a pretty important piece of that for determination of injury and potentially for quantification too. So let's see, I don't want to go too far over on time, but I think that is all I've got here. Thanks for your attention, and I'd be happy to answer questions if we have some more time.
Thank you, Jeff. We have about 1 minute. Is there anyone who had a really quick question for Jeff?